Friendly's making like $150 every donation. Really? Yeah. At least for like the first 8 donations. I mean, they sort of slowed down, but that's fine.
I heard that they might give you a tax credit. Actually,
Really? I could use
that
that.
they might pass that. Yeah. Yes, so we're gonna finish up transaction. Practice questions and then we'll move on. Yeah. Okay, happy Monday. Our next exam is a week from today. So I don't think we'll quite finish up our genetics stuff today. We may likely have to go into Wednesday a little bit, but I anticipate that most of Wednesday's lecture on viruses, that will not be covered on Monday's exam. So I originally thought we were going to finish today, but I don't think we're quite going to make it. So just kind of be thinking about if you haven't started that homework, it's time to be doing that. Definitely the metabolism and the genetics, in my mind, is more complicated than the eukaryotes and the growth stuff that we did. But we'll start with chapter 5, the eukaryotes. That was chapter 5, then chapter 7, growth. Metabolism is 9, and then 10 and 11 here are genetic stuff. So, okay, so I'm anticipating more traffic in my office as the week goes on. I am available by Zoom tomorrow morning only. Thursday and Friday, my schedule's a lot more open, and I can meet with you. So take advantage of that. Where we left off last time, we almost got through transcription. I'm just going to finish up a couple more points on transcription, and then I'm going to give you a practice question. I want to use that practice question to kind of review transcription a little bit. So we finished up last time thinking about how transcription ends. There's 2 different ways that we say the making of the RNA, we're only going to be transcribing a certain portion of the DNA. So we talked a lot about how that starts, a couple different ways that it stops. I want to point out a few differences. Always assume as we're walking through these processes that we're talking about prokaryotic processes, but I want to show where there's some differences between how we do it in eukaryotic organisms than prokaryotic organisms. First thing, complexity. It's a lot more complicated in eukaryotic organisms. There's things, I think they're proteins called transcription factors that have to be there for initiation to take place, even for the elongation process to continue. So there's a lot, it's just a lot more complicated. Eukaryotic transcripts are monocistronic. So that means that mono meaning one and cistron meaning gene. So for each transcript, that's the information that's going to be translated into one protein for eukaryotes. Prokaryotes can have polycistronic transcripts. Which just means that one transcript or one messenger RNA can encode multiple proteins. Okay, so that will become more obvious as we talk about regulation and how operons work. But just wanted to point out those terms, monocistronic versus polycistronic. There is some processing that usually will take place in eukaryotic messenger RNA after termination takes place, because remember that transcription in eukaryotes is taking place where the DNA is in the nucleus. So in order for that messenger RNA to get to the ribosomes, it has to do some traveling. It has to leave the nucleus. So in order to stabilize that RNA, they kind of add a little bit to the ends to protect the ends. Okay, there's a 5' on the 5' end. We have what's called a guanine cap. And these are nucleotides with a little modified guanine base. They have some little different linkages and some methylation that takes place. But a 5' guanine cap and a 3' polyadenylation. Okay, so here's the messenger RNA on the 5' end. There's lots of guanines and the 3' end polyadenylation. Okay, then the ends are not so subject to being chewed up by enzymes, endonucleases within the cell. So that's going to stabilize the RNA. In addition, the messenger RNA is going to need spliced because it's going to include both introns and exons. Okay, the introns or intervening sequences are going to be cut, and then the exons, or you could think of them as expressed genes, are going to remain and get translated into proteins. All right, so sometimes, which is capped to the introns or the exons, if you can think about expressed genes, exons, and that helps you remember. So this graphic from our old textbook, I really like it. It kind of puts some of those things together. Okay, so here's, again, eukaryotic messenger RNA. Here's the template DNA strand, the one that's used to make this temporary messenger RNA, okay, during transcription. Here's a 5' cap, the poly A tail, got exons and introns in there. The introns need to be cut out. So you have this big protein complex called a spliceosome complex that goes through and gets rid of those introns. So now what you're left with is just the introns, and that's what leaves the nucleus and travels to the ribosome, and it will get translated. Okay. Here's another way to show that splicing. This is kind of one way that some regulation can take place. There's actually different ways that a transcript can be spliced. If here's the DNA, and I think the exons there are the squares, and then the lines in between are the introns. So if just the introns were spliced out, you would expect this messenger RNA. If it was spliced maybe a little bit differently, I think this bottom one here, where that yellow exon is gotten rid of, you would get a little difference. Messenger RNA, and the one on the right here, I think, is the upper one here, if part of that one was cut off. So you could get actually 3 different proteins depending on how the splicing takes place. So we think that it can control that a little bit. Sometimes errors are made in splicing, and we know that some diseases in humans we attribute to errors in splicing, like Duchenne's muscular dystrophy, possibly Parkinson's, maybe some other ones like cystic fibrosis. So, okay. All right, so let's do a practice problem. It's one of those kind of wordier ones, but whoops, boy, that's All right, so read through that, check what you think is the best answer, we'll talk through it. All right, so correct answer we're looking for here is B. All right, so B is correct. So let's go through and talk about why the other ones are not correct. E here, transcription takes place in the nucleus. That is true for eukaryotes, not prokaryotes. So we gotta make sure you read that clearly. Of course, prokaryotes don't have a nucleus. D would be correct if it was RNA polymerase. Okay, DNA polymerase 1, that has a role in replication and getting rid of the primers, those temporary RNA primers. We talked about this, more errors are made actually during transcription than replication. We want the blueprint to be correct. So we talked about how the error rates of RNA polymerase is a little bit higher than DNA polymerase 3, which is the main enzyme in replication. To address these two, let me just go back to initiation here. I think I said, oops, the -10 region is downstream. Why did it do that? They got spliced in there somehow. I don't know how that happened. Okay, but you remember this slide, I think one of my answers was the -10 region is downstream. So the start site is is like right here. And then when this is upstream, downstream is when transcription begins. And then the correct answer was that there are sequences that are going to depend on how strongly RNA polymerase binds to the DNA. Okay, so there's this -10 region, -35 region. We talked about how closely those sequences of bases are to some consensus sequence. So that was where this slide came in, and some consensus sequence. We found that when a certain base is closest to this consensus sequence, then the RNA polymerase is more likely to bind. Okay, so it's kind of written into the genotype to some extent, how likely RNA polymerase will bind. Sigma factor also helps RNA polymerase bind to that promoter region. Okay, questions there? Okay, well, oh, yeah. So here I'm talking specifically about prokaryotes. There's a lot of similarities between eukaryotes. Some of it is just a little bit of terminology, and I think in eukaryotes, I'm not as familiar with how it happens in eukaryotes, but it's more complicated. Okay, so yeah, assume that this is bacteria and archaea only. All right, we'll do another transcription question here shortly, because I think generally, I think generally the transcription, the making of RNA from DNA somehow is a little bit more complicated. But let's move on to translation. So this is how that messenger RNA transcript gets changed into a language of amino acids. It's translated into proteins. But before we do that, we have to have a little bit of an understanding of the genetic code. So where did this genetic code come from? Do I have to memorize it? No, you don't have to memorize it. We figured this out through a lot of trial and error back in the 1960s. A guy by the name of Nuremberg was working at the NIH. A lot of trial and error, a lot of radioactive labeling to try to figure out which nucleotides were getting translated into which amino acids. Okay, so again, we call this the genetic code, and it takes into account codons of messenger RNA and basically which amino acids they will correspond to. Okay, so you take the first letter of the codon messenger RNA, GAC or U, the second letter, and the third letter here. Notice that there is some what we call degeneracy or wobble. In the third base pair. Okay, so like in this column here, it doesn't matter which that third base is of the messenger RNA codon. It's still going to correspond to the same amino acid, serine. Now, sometimes it does matter, like here, if it's U or C, it's going to be phenylalanine. If it's A or G, it's going to be leucine. So, but there's still, again, sometimes that third base in the codon, it doesn't really matter what it is. I'd also call your attention, AUG, that's the start codon. That's always going to be at the beginning. You find that at the beginning of a transcript. In bacteria, that will encode for formal methionine. In eukaryotes, it's methionine. So for some reason, the bacteria add a little formal group on there. And then there's 3 stop codons. And again, I don't need you to memorize that, but those do stand out a little bit. When those are encountered by the ribosome, then that's what's going to stop translation and cause the cessation of the making of the new protein. Okay. All right, so if we look at the steps of translation, you can see we kind of split it up the same way as transcription. We have some initiation, elongation, termination. But before initiation, often like to think about the charging of the tRNA. And I'm not going to go through the steps of how that happens, but that just basically means that some amino acid is attached to the tRNA. And there's basically one enzyme that facilitates that. It costs some energy to do it. And the amino acid is attached kind of up there at kind of the top of the graphic there. Which amino acid is put on, it does make a difference. It depends on the anticodon of the tRNA. Of course, that anticodon is going to be complementary to the codon on the messenger RNA. And there's other kind of things that help the enzyme kind of understand which enzyme to put on there. But a charged tRNA is one that's carrying an amino acid. And let's see, and then, all right, let's talk about how the process gets started. So I've got a couple different ways to show you this. These are some pictures from our old textbook, and then I'll show you a different one. So here's the small ribosomal subunit. And we've got the messenger RNA here that's docking on that small ribosomal RNA subunit. We'll talk a little bit more how it's really important that that docks in just the right spot. The first tRNA that's going to come in and start the protein, okay, it's always going to be carrying that formal methionine amino acid for prokaryotes. And the anti-codon of the tRNA is going to match up with the start codon AUG. And that's, we have a specific name for where that happens. We call that the P site. P stands for either peptide or peptidal. Either way, P for peptide, that's where the growing peptide is going to be anchored. Okay, so the first one comes into the P site. As we'll see, all the other ones are going to come into this other site. Sometimes we refer to this as the acceptor site. I think your book calls it the amino acyl site. Okay. A stands for either one of those things, so either way. Okay, so some energy in the form of GTP has to be spent. Why is it GTP? Why is it not ATP? I don't have a good answer for that. It's just the currency that's accepted by the making of proteins. It's very similar to ATP. And when that first amino acid charged tRNA docks there at the small ribosome, only now does the large subunit of the ribosome come to make what we call an initiation complex. Okay, so now it's kind of like everything's all in place and ready to kind of elongate the polypeptide. All right, so I like this next slide before we get into elongation. A couple more details that I want to show, and this came from a different textbook. So here's the messenger RNA down here. And this picture shows the small subunits on the top of messenger RNA. Okay, just so this small subunit of the ribosome, this is a piece of the ribosomal RNA in the small subunit of the ribosome, the 16S piece. And in that 16S piece of ribosomal RNA, there's a spot, a particular sequence that's going to bind to the messenger RNA. Anytime we have a kind of a special name for something, we know it's important. And there's a special spot on the messenger RNA that binds to that 16S ribosomal RNA, and we call that the Shine-Dalgarno sequence, or the Shine-Dalgarno element is what they call it here. Okay, so this is its job. It binds to the small subunit of the ribosome. And it's really important that that lines up exactly, because as long as this lines up exactly, then this AUG is going to line up exactly with the P sites. Okay, and if that was lined up, maybe not quite exactly, maybe it was lined up like this, that's going to make a big difference. That's going to read it in a whole different reading frame, sets of 3. So like I said, it's important that that gets lined up just right, so it's reading in the right reading frame. Another thing I want to point out, it's going to kind of start at the 5' end. And that's not something you need to memorize. You can kind of think through that, because in prokaryotes, transcription may still be taking place. Okay, the DNA is out in the cytoplasm. All right, and we've got some RNA polymerase here. This is my rudimentary drawing. RNA polymerase maybe is still transcribing the DNA, and at the same time, translation can begin as transcription is still going on, because it's all kind of in the same place. For eukaryotes, that DNA is in the nucleus, and that transcription has to finish before it can come out into the cytoplasm. So, and remember, the new ribonucleotides are always being added on to the 3' hydroxyl group. Okay, so you know that's the 3' end, so the other end must be the 5' end. So And in fact, we'll see this a little bit later for eukaryotes, it's the 5' cap that actually will line up with the ribosomes. They don't really have a Shine-Dalgarno sequence for eukaryotes. Okay, so this is for prokaryotes. So we've got the initiation, we've got that first tRNA come in. Now, after the first one is docked, all the new charged tRNAs are going to enter at the A site. Okay, so let me write that. After initiation, all new tRNAs dock at the A site. Okay, and then remember we said that ribosomal RNA can act like an enzyme, and when it does, we call it a ribozyme. So there's ribozymes in that large subunit that are going to act to catalyze the peptide bond formation. That really briefly sits in the A site, but then it very quickly moves over into the P site. So we say that the growing peptide sits at the P site. All right, and I should maybe put sits in quotes because this process happens so quickly that it's not really sitting around. I think 15 amino acids are added per second. Okay, so kind of mind-boggling how quickly all the pieces can get put into place here. And then if you go another round, okay, here's a new charge tRNA coming in. It's going to dock at the A site. The growing peptide does get transferred to the A site, but very quickly, you know, whether you think about the RNA moving through there, kind of like ticker tape or something, that growing peptide, again, is going to be sitting at that P site. Once this tRNA gets rid of its amino acid, it moves over to this E or exit site, and then gets released and maybe gets charged up again. This is a really nice video that I found on YouTube. I'll just maybe show you a little bit of it, and probably I don't have figured out how to show sound in here, so you can kind
of how
maybe watch it later and the narrator voice a little bit. It does show transcription. I'm going to move it till
it snakes
there's
away
the
from the nucleus.
this
and
is actually in
into the outer part of
eukaryotes,
the
but
cell.
there's a transcript leaving the nucleus. So that's how you know
Then,
that
in a
it's
dazzling
actually
display of
eukaryotic, but
choreography, all
that's
the
okay. So
components
there's the
of
messenger
another molecular
RNA.
machine
There's a
locked
small
together
subunit of the
around the RNA
ribosome.
to form a miniature
That's
factory
clamping
called a
on,
ribosome.
and there's the large subunit.
It
Now the
translates the
messenger
genetic
RNA is
information in the
moving
RNA
through the
into a
ribosome.
string of
There's the
amino acids that
tRNAs
will
coming
become
in.
a protein.
And you'll see the growing polypeptide in a minute. It's going to be
Special
red.
transfer
There's
molecules,
the
the
tRNAs.
green
The amino acid
triangles,
is just a little
bring
red
each
tip
amino
at the top.
acid to the ribosome.
So again, 15 amino
The
acids
amino
per
acids
second.
are the small red tips attached to the
How
transfer
do they
molecules.
find the right thing? How's the right one go in the right place?
There are
Really
different transfer
astonishing.
molecules for
Okay,
each
there's
of the
different
20
amino
amino
acids, right? There's like 20 different amino
They
acids.
all carry a specific 3-letter code that will be read by the machine. Now we come to the heart of the
There you
process.
can see the inside.
Inside the
Okay, and then
ribosome,
the little
the
red string
RNA
growing
is
at the
pulled
top,
through
that's the
like a
growing
tape.
polypeptide.
The code for each amino acid is read off,
Okay.
3
So
letters
eventually,
at a time, and matched
a
to 3 corresponding
normal
letters on
or
the
an
transfer
average
molecule.
average protein has about 300 amino acids.
When the right transfer molecule
So
plugs
eventually,
in, the amino
talk
acid it
about
carries is
termination
added
yet
to the
here,
growing protein
but
chain.
eventually the ribozymes will
Again, you are
cleave
watching
off.
this in real time.
That's growing
And
polypeptide
after a few
when a
seconds, the
stop
assembled
codon is
protein
read.
starts to emerge from the
Okay, so I
ribosome.
think you get the idea there. Let's go back. Okay. This figure, kind of like what I was trying to draw in before. Just again, this is prokaryotes, where the DNA can be transcribed kind of over and over and over again, and you can have this polyribosomal complex where you've got different ribosomes just swooping right in on that same messenger RNA transcript. The messenger RNA gets degraded fairly quickly, so this can't happen indefinitely, but you can definitely have, you know, proteins, that same protein sequence translated over and over again. Okay. All right, so how does protein synthesis terminate? When a stop codon is read on the messenger RNA, so here's one of them here, UAA, there's kind of a pause because there's no tRNAs that are going to match that. So proteins called release factors will come in and bind to that messenger RNA and cause the cutting of that polypeptide, again, by ribosomes. The ribosome is going to then, the complex is going to dissociate. Okay, back into the small ribosomal subunit and the large ribosomal subunit. Okay, write this in, ribozymes. I'm going to kind of sever that polypeptide chain. So now we have a workable protein. Well, maybe not quite yet. Because sometimes there's some modifications have to be made in order for that protein to be functional. Usually for bacteria, that formal methionine is removed right away. And there might be a lot of different reasons why that happens. One reason is that identifies that protein as being of, you know, bacterial origin. So it may be, if it's a bacteria that's trying to, you know, infect some kind of host cell, it might not want that, you know, kind of red flag that this is not part of the host's genome. If this is going to be an enzyme, then there might be some coenzyme or cofactor, some non-protein part that's necessary for that enzyme to function that might have to be added. There might have to be some folding or some bonding to another polypeptide, if it's an enzyme, for that to function. Sometimes there have to be some cuts made. We see this in some proteins that get made by viruses. They have to be cut. There's proteases that have to come in and cut these proteins so that they're functional, and we might be able to have drugs that act on those proteases to keep them from working. But a bacteria may not want that protein to be functional right away. If it's something like a toxin, you don't want that to be functioning inside the bacterial cell. You want it to be functional right when you let it loose. So I'll just some modifications that potentially have to be made. Okay, so I find that a lot of students have a little trouble, at least in first, not mixing up transcription and translation. They kind of sound alike. So try to keep them separate in your mind. Let's have another practice question about transcription. Okay, which one sounds right? It's only one. Actually, E. E. Okay, so let's walk through it. So I mentioned that briefly before. In initiation of transcription, these sigma factors will come in, bind to RNA polymerase, and help it bind more tightly to the DNA. And the more tightly the RNA polymerase binds, the more likely transcription is going to take place. Let's see, why are the other ones wrong? So in transcription, only one DNA strand or the other is transcribed. Okay, potentially they both could be, but that would be kind of an unusual situation. It's certainly, again, the one that's transcribed is going to be the template strand. This would be correct if it was translation. Okay, so again, you have to read them closely. We talked about that. Initiation of translation begins with that Shine-Dalgarno sequence on the messenger RNA lining up with a small subunit of the ribosome. This one, DNA helicase unwinds the DNA strands. RNA polymerase unwinds the DNA strands itself. Okay, DNA helicase is necessary for DNA polymerase 3, which is the enzyme of replication, to start copying the DNA in replication. All right, so you can see how you got to keep replication separate from transcription, separate from translation. And this is true. Replication begins at a spot called the origin. So then it would be true if it said replication. So in replication, the whole chromosome just gets copied, and that starts at a place called replication. So what do I mean by unwinds the DNA? You've got two strands of DNA. The base pairs are complementary to each other, and another way to say that is unzipping. When those hydrogen bonds between the complementary strands are broken. That's the DNA helicase. Now we talked about another enzyme, DNA gyrase, that deals with the supercoiling that can take place when you're continually, opening up the replication fork. That happens upstream. It kind of relaxes that supercoiling, clips it, relax, turns it around, and seals it up again. Okay, but it's the helicase that pulls the strands apart. Yeah. Well, so DNA polymerase 3 can begin replication. Yeah. C. So if C, well, depend. If you're talking about, if you were going to make it talk about replication, you would say DNA helicase unwinds the DNA strands, so DNA polymerase 3 can begin replication. If you're still talking about transcription, basically RNA polymerase doesn't need anything to unwind the strand. It does it itself. It kind of has multifunctional enzyme. It's good to practice on these minutia here. Good. All right. Oh, and so here's a table that is helpful. Just cover it up. See if you know what it's talking about. It's kind of just the basic idea. We talked about everything except we didn't talk about the directionality of translation, so you can not worry about that. I think sometimes the direction of polymerization, sometimes that's very confusing because it's unclear. Are you talking about the direction on the strand that's already there or the new strands? Okay, the new strand is always going to be synthesized adding on to the 3' end. Okay, so we say that 5' to 3'. Okay, so it's always adding on to the 3' end of the new strand. That's always confusing. So again, cover that stuff up, know what the enzyme is talking about, just so that's just kind of cover the basics there. All right. So it's in the best interest of the cell only to make the proteins that it really needs. And there's different ways that a cell can kind of regulate the production maybe of those proteins that it needs. So we're going to talk about that next. Why does it want to conserve energy? Well, it's very expensive to make proteins in a cell. Any guess as to how many ATP it takes just for that polymerization reaction of translation, adding the new amino acids onto that chain? For an average protein of 300 amino acids, what's your guess? How many ATP? 30. Okay, I will tell you that's a little low end. Let's go a little higher. 300. Okay. What's that? 600. You're getting closer. Somebody last semester said 1 million. Okay, no, not quite that much. Okay, so you're in the right order. About 1,200 ATP to make a protein. And I tried to double check that number before class, and you kind of read different things, different places, but that's about kind of in the neighborhood, a couple ATP here for this reaction, a couple for that. So, it's expensive, right? And if you think about a cell, maybe a bacteria cell that's only doing fermentation and only getting, a couple 4 ATP from every glucose. So yeah, it doesn't want to make proteins that it doesn't absolutely need to make. So we talked about some different kinds of regulation already. We've mentioned this consensus sequence variation. So that goes back to this slide. And, depending on looking at the promoter region, if that's, base is one base is a little different, the RNA polymerase will bind as well. You won't as often get the protein. So that can be kind of written into the actual sequence, how often a protein gets made. You can use, a cell can use different sigma factors that will help the RNA polymerase bind more or less tightly. Okay, so those are both kind of at the level of transcription. Another kind of regulation at that level of transcription is the operon. And this is just when genes are regulated together. And it's usually for proteins that are in the same pathway. So it's a very efficient way if you need, say, these 3 enzymes to synthesize something in the cell. It makes sense to regulate those 3 enzymes, that you would need the same amount of all 3 of them. So just regulate them together. So it's a very efficient way for prokaryotic cells to regulate what they need. We think this is primarily a prokaryotic thing. So bacteria and archaea. Eukaryotes have some kind of physical clustering that may be kind of similar to that, but it's really not the same. So when we talk about operons, this is primarily a prokaryotic thing. Okay, so don't forget everything that we just talked about transcription. Okay, because we're still talking about transcription here. So if you think about kind of the parts, and I know that you've talked about operons in some class at some time before this. We're talking about the DNA, and remember these are base nucleotides in DNA. There's a region of the DNA that we label the promoter. That's where the RNA polymerase binds when transcription takes place. A little bit upstream of that is the operator. This is where a repressor will bind if there is one involved. That repressor protein gets made at a little different spot on the chromosome. That's where, you know, some other spot where some repressor might be made. And then here's the structural genes down here. So for example, maybe those structural genes get transcribed and then translated into 4 different proteins that either are necessary to make something within the cell, or maybe they're necessary to break down some nutrients that the cell may be able to use to get energy. Okay, so we're going to look at 2 different operons, and hopefully, as 2 examples will help kind of trigger your memory. Okay, so there's 2 different kind of ways that operons kind of control that they can fall under. If they fall under negative control, that just means that there is some repressor that can be present. Okay, and of course, like I said, the repressor binds to the operator, because if you've got something bound here to the operator, then the RNA polymerase, it's going to be stopped if that repressor is bound at the operator, and it can't transcribe the gene. Okay, so it makes sense that's where the repressors bind. There's 2 types of negative control. There's induction and there's repression. In an inducible operon, that just means that transcription is usually not taking place. Okay, so we'll say it's usually off, meaning the DNA is not transcribed. Okay, so in an inducible operon, there's some kind of inducer that has to bind to the repressor to get it to come off of the DNA. And most often, catabolic pathways are regulated this way. So catabolic pathways are things like the lac operon, where there's some catabolite, there's some like nutrient, some sugar, and then the cell needs certain enzymes to break down that sugar. In a repressible operon, okay, these are usually on, meaning the DNA is usually transcribed. And they're only going to be stopped from being transcribed if there is a repressor that becomes activated. All right. So anabolic pathways are often regulated that way. So the example that we're going to look at is the trip operon. This is a biosynthetic pathway. This is how the cell makes tryptophan, an amino acid. Okay, so that might not be as familiar. Enzymes necessary to synthesize that amino acid tryptophan. So it's always best to try to think through this instead of like memorize what happens. Try to think like a bacteria. So try to think what the bacteria needs and what it doesn't need. So if we look at the lac operon, okay, so this is remember, this is a DNA, and this is a gene for the LAC-Z, LAC-Y, and LAC-A. Basically, those are enzymes. That's the DNA that's going to encode for the proteins, for the enzymes to metabolize lactose. Okay, LAC-Z is actually encodes for beta-galactosidase. That's the enzyme that splits that disaccharide lactose into glucose and galactose. Okay, if you can't split it, then you can't use it for an energy source. The LAC-Y is a permease enzyme, so lactose can come into the cell really quickly. It can come in a little bit on its own without that, but it's kind of like a fast pass. You know, like you're driving down the road in Chicago, you got your fast pass, you don't have to stop and pay the toll. Okay, so without the permease enzyme, it's slower for lactose to get in. And the LAC-A probably has some detoxification function. It's not really clear what that's necessary for. Okay, but so the cell only wants to make these enzymes if there's actually lactose present. There's no sense making these enzymes to metabolize lactose if they're not, if there's none available. Let's see, in the absence of lactose, there's going to be a repressor here that's bound to the operator. Okay, and that's a physical block. The RNA polymerase can't transcribe that gene. Even no matter how well it binds to the promoter, it is not going to get past the operator to transcribe those genes. Okay. But in the presence of lactose, lactose is going to bind to the repressor to disengage it from the DNA, and now the RNA polymerase is going to be able to transcribe those genes that are necessary to metabolize lactose. Now it's actually a little more complicated than that. We'll add another layer of regulation on there in just a minute. Okay, let's look at a second operon, the trip operon. And like I said, with the trip operon, the structural genes here are going to encode enzymes to synthesize tryptophan. I'll abbreviate that because that's hard to spell, tryptophan. Okay, so normally the repressor is off here. Tryptophan, it's amino acid that's the cell needs for lots of things to make lots of proteins. Okay, so here in the absence of tryptophan, the cell needs tryptophan. So it's transcribing those genes that will be translated into those proteins. When tryptophan is present, though, then tryptophan is going to bind to the repressor to activate it. It's kind of like a co-repressor. And now it's bound to the operator and RNA synthesis is blocked. Okay, so if we say grow our E. coli in a medium that already has tryptophan in it, the E. coli doesn't want to keep making tryptophan because it's already there. It doesn't need any more. Okay. So it's going to save ATP. All right, so there's one, I want to go back to the LAC operon a minute here. And it's actually just a little bit more complicated because if there is lactose present, but there's also glucose, the cell doesn't really want to worry about using the lactose because that's more complicated. If steak is your favorite and you get offered steak and chicken, you're not going to eat the chicken first, you're going to eat the steak first. So glucose is like steak to the E. coli, and it wants to use the glucose first. So even though lactose is there, it needs to have a way to repress using lactose, or it doesn't want to make those enzymes to use lactose. So it wants to just use the glucose first. So the evidence that we have that there is another layer of regulation in there is when we do a growth curve, when we're growing, say, E. coli in some medium with both glucose and lactose. And maybe over time, we're taking that culture out and getting absorbance readings from a spectrophotometer, and we make a growth curve. And what we see is we see two different lag periods. And we call this dioxic growth. It's just a two-phased growth. Right? A normal growth curve, right? You have your lag period, and then you have your log period, and then your stationary phase, right? So log is when it's when it's growing exponentially, growing really quickly. But we don't see that. We see 2 lag periods, and we see 2 kind of separate log periods. Because the organism is using the glucose to begin with, and then when the glucose runs out, there's another little lag while it ramps up its lactoferrin and makes those enzymes that it needs to metabolize lactose. Okay, so we call this catabolite repression. So metabolism or breaking down of a catabolite, lactose is a catabolite, okay, so metabolism of lactose is halted or repressed so that glucose can be used. All right, so the mechanism of how this happens falls under what we would consider a positive control. So this is the best example of some positive control mechanism that I could give you. And that involves a molecule called cyclic AMP. Cyclic AMP is related to levels of glucose in the cell. It's actually inversely proportional. So when glucose levels are high, okay, then the cyclic AMP in the cell is going to be low. Okay, the converse is true. When glucose levels in the cell are low, then we're going to have lots of cyclic AMP. Okay, so if glucose levels are low, there's lots of cyclic AMP. It's going to bind to this catabolite activator protein or CAP protein. We have this CAP cyclic AMP complex, and that is recognized by RNA polymerase then as it has to recognize, the RNA polymerase has to recognize it so that the lactoferrin can be transcribed. And before I think I stuck this in here just because I had room for it. This is about a 5-minute YouTube video. It's the best one I've found of just the lactoferrin. It's not flashy, it's not entertaining, but it goes through the lactoferrin and then the positive control of the lactoferrin as well. So here's your book's figures walking through this. So in the absence of cyclic AMP, the CAP doesn't bind the promoter. You get transcription maybe just really slowly if that activator complex isn't there. You might still get a little bit of those enzymes to break down lactose. But if glucose levels are low, doesn't say that there, then cyclic AMP is going to be high. That is going to be right there. That complex right there at the promoter region, RNA polymerase recognizes it and now will transcribe the genes to metabolize lactose. Okay, so that's what that's showing up here at the top as well. But remember that even if that is there, and if there is no lactose, okay, then the repressor will still be bound and you won't get transcription. So there has to be lactose presence and low glucose in order for those genes to get transcribed. Okay. Okay, a practice question. Okay. Okay, so the first one's not right because it says the repressor stays bound to the promoter. Remember, repressor binds to the operator, so you can cancel that one out. There is lactose present, so that's going to bind to the repressor and release it, so that's not true. Correct answer is here, lactose, which is the inducer, or sometimes it's allolactose, which is really similar to lactose. Deactivates the repressor, and then the RNA polymerase can transcribe the gene. Okay, so this says for answer D, lactose binds to the promoter. No, that doesn't happen. That one's not true either. So, okay, questions? Remember, try to think like what the cell needs and why it needs it, and that really makes more sense. So thinking about how genetic variation can take place. All right, bacteria, when they divide, when they grow, they're using binary fission. They're just, each daughter cell is basically clones of the mother cell. So how can any kind of genetic variation ever take place? Well, one way is by mutation. Another way is by horizontal gene transfer, which we probably will finish that up next time. But mutations typically are going to be bad almost every time, but every once in a while you might get a mutation that actually helps a cell survive better. We're just going to kind of dabble in this. We're going to talk a little bit about point mutations and a little terminology there. But a point mutation is just when a single nucleotide base is affected, maybe the wrong one inserted. So a guanine instead of a cytosine, for example. If you get an extra nucleotide inserted in the DNA, or maybe one accidentally deleted, that can lead to frame shift mutations. And those are typically pretty serious, as we'll see. So we want to be able to identify these different kinds of mutations, silent, missense, or nonsense. All right, so let's look at examples of each of these. So, here. All right, so say you have a DNA strand up at the top here. Look at the top one. And then this strand right here, it's supposed to be an adenine base, but say it gets changed somehow to a thymine. We'll talk about different kinds of mutagens in a minute. You can see that the amino acid sequence here is the same because of that wobble in the third base of the codon. Okay, so that didn't make any difference. So we call that a silent mutation. It doesn't have any effect. We still have the same sequence of amino acids that make up the protein. What if, let's see, this base right here, this cytosine was changed to an adenine, then you would have you would have ACC instead of CCC, so you'd have a threonine amino acid instead of a proline. Okay, so that makes sense. It encoded a amino acid, but it wasn't the right sense. Okay, so we're going to call that a missense mutation. And that might make a huge difference as to whether the protein was functional. It might not make any difference at all. It just depends where that amino acid was in the protein and whether it was in an important place. What if the last one here, what if this C was changed to a G? Now, instead of tyrosine, that's going to encode for a stop codon. All right, so that's generally going to be pretty bad, especially if it's right at the beginning of the polypeptide. It's going to not translate the entire rest of it. Maybe though, if it was at the very end, maybe that wouldn't have that much of a difference. But we refer to that as a nonsense mutation if a stop codon is inserted instead of an amino acid. And then a frame shift mutation here, that's just if you have typically some kind of insertion or deletion. So here it had this T and A removed. So after that, it's a whole different reading frame. And after that, all the amino acids are going to be different. So that's typically going to be a pretty serious mutation. The protein's probably not going to be functional unless it was at the very end of the polypeptide. All right, so no difference between that terminology, missense, nonsense, silent. We'll take a minute here for a question. I have this one in here. All right, so number 3 here, most likely, because the 5th codon, that's right at the beginning. If there's going to be 300 codons in your protein, that's right at the beginning. So everything after that 5th codon is going to be different. So it's likely to make a huge difference. If it was a nonsense, a stop codon at the very end, maybe not too much of a difference. It's, again, a different amino acid, maybe just one place, not as much as lots of them being different. And of course, it's a silent mutation. It doesn't matter where it is, it doesn't do any different. So, okay, what kind of things cause mutations? A lot of things. Let's talk a little bit about radiation as well, and then we'll mention a few chemical mutagens. So radiation can either be considered ionizing radiation or non-ionizing radiation. So if you look at, I'll come back to that in a minute. We looked at the spectrum of light before when we were talking about fluorescent microscopy. Remember fluorescent microscopy, we were using wavelengths in the UV range. Okay, that is considered non-ionizing. And that's somewhere between about 10 and about 300 nanometers are typically considered in the ultraviolet range. It's a little bit, it's not a specific wavelength because what we considered non-ionizing radiation, it depends on the effects on the cells. So it's about 10 nanometers considered non-ionizing. And then below that, these are ionizing wavelengths, shorter wavelengths, higher energy, X-rays, gamma rays. Okay, so they cause different kinds of mutations. Ionizing radiation, again, X-rays and gamma rays, they will cause lots of different problems in the cells, but here we're looking at the effect on DNA. It can cause single or double-stranded breaks. Okay, so the backbone of the DNA, the sugar phosphate backbone, the ionizing radiation maybe can break it in one place or in both. One strand sometimes can be repaired. It's a problem when both strands are repaired because then there's no template to work from. Okay, so it can be pretty severe mutations. Non-ionizing radiation causes a specific kind of mutation called a thymine dimer, where 2 thymines on one strand will actually bind to each other on the same strand. So they're only supposed to bind to the complementary strand, not to each other. Why thymines? It's just the nature, the structure of the thymine that makes it, that base that makes it more likely to happen. Sometimes cytosines, so it's some kind of pyrimidine, either cytosine or thymine, more likely with thymines. And that causes a kind of you don't really show it very much here. They say kink. It causes a little bit of an outpocketing of the DNA when those 2 thymines bind to each other. So if RNA polymerase 3 was going down to copy that strand, it probably wouldn't read it correctly, probably make mistakes. If RNA polymerase was was going down that strand to transcribe it. Probably wouldn't transcribe it correctly. Okay, so luckily, a lot of cells have ways to fix, especially thymine dimers, because that's a very common mutation because, UV, it's in sunlight, it's just a very common mutation. Okay, so there's some examples of radiation that can be cause mutagenesis. And we actually will use radiation to do that on purpose when we're trying to control bacterial growth. We'll get to that in a couple weeks. Here's a few examples of chemical mutagens. And one particular kind that I like to talk about are the nucleoside analogs. Remember what a nucleoside is? That is the base, adenine, thymine, guanosine, cytosine, as well as the sugar. Okay, we call it a nucleotide if it includes phosphate. So these are analogs of nucleosides. That means they mimic, or they're really structurally similar to a nucleoside, but they're not exactly the same. So if they are incorporated into a DNA strand, they won't act the same way. So here's an example, AZT. You might recognize that was the first drug that we had to treat HIV/AIDS. And azithromycin. So here's a structure of azithromycin. It has the base thymine, the deoxyribose is the same, except it has this azide group here instead of the 3' hydroxyl group. So because of this azide group, when HIV incorporates azithromycin into its DNA, it doesn't have that 3' hydroxyl group to continue the polymerization reaction. The replication stops. So it lacks that 3' hydroxyl group. So it was, fairly effective. It's one of the drugs of many that we have now to try to control the replication of HIV. It goes by its trade name now, which is not as familiar to, I don't know, me anyway. Another example, here's a chemical 5' bromouracil. Instead of halting chain elongation, it actually introduces mutations because it binds to guanine instead of adenine. So here's the structure, like just ignore this. We'll just skip over that. But here's 5 bromouracil. You can see that structurally it's very similar to thymine. It just has a bromine here instead of a methyl group. So that gets incorporated into DNA strands, but then you can, so right here, it's say it's incorporated, but it binds to guanine, where if that was thymine should bond to adenine. Okay, so it's complementary to a different base, so you get lots of different mutations that are going to build up because say this new strand that's being made is not the right complementary basis. Another example, a little closer to home, about a year ago, January 2022, our first drug used to treat COVID-19 that we could take as a pill form was approved by the FDA. And it was approved by a really narrow margin. They have committees that decide these things, and it was like 13-10. They decided to approve it. The reason that it was such a kind of a controversial thing among that committee, whether to approve it or not, is because this molnupiravir, it's a cytidine nucleoside. So here's cytidine and here's molnupiravir, a few differences there. But the idea is that the COVID-19 virus would incorporate molnupiravir in, and it would cause what they call catastrophic mutation. So not a chain termination, but because it would, again, pair with a different base pair. And that would cause, COVID-19 to not be replicate as quickly. The controversial thing was that, hey, do we really want to increase the mutation rate of COVID-19? it's already mutating quickly enough. So it was approved, but not for pregnant women. And luckily, a week later, we had a better drug that was approved called Paxlovid, which we'll talk about more later, that works by a different mechanism. So enough for today. I anticipate maybe 10 minutes or so into next period. So have a good Monday.